Good morning, everyone. Welcome to the fourth session, which is on implementation of genomic medicine in different settings. So myself and MK will co-chair this. I will introduce the first two speakers and MK will take over after me after that. So the first speaker is going to be Alan Shuldiner. Alan is, as you know, the co-chair with me on the genomic medicine flagship project on rare disorders. He's also with Regeneron Genomic Center and at the University of Maryland. So over to you, Alan, to talk on the G2MC's Genetic Clinics for Rare Disorders flagship project.
Well, thanks so much, Vijayra. It's a pleasure to be here and an honor to present on behalf of the co-investigators of this pilot project. While the first slide is coming up, I should say that everyone in this room should actually be very proud of this project in that it was born here at G2MC meetings, and the collaborators that and participants of this project, I know I would have never met if not for G2MC, let alone engaged in this amazing collaboration. Okay. So this is the team, the G2MC Diversity Rare Undiagnosed Disease Flagship Program. And as I said, these are individuals I would have never met if not for G2MC. And I guess it was born out of the idea that, you know, these conferences are wonderful and we exchange ideas and information about implementation of genomic medicine. But at the end of the day, people have to be doing things, and G2MC being a flagship for this initiative and effort, we felt it was really important to begin these flagship programs to actually implement and make a difference for patients in low and middle income countries. So in terms of an outline today, I'm going to tell you a little bit about exome sequencing for rare undiagnosed diseases. Not everyone in this room is totally familiar with this indication for molecular testing. I'll tell you about the G2MC pilot study design. If I have time, I'll present a couple of individual case examples to make this real and personal for all who are listening. Summary of the findings, lessons learned, and the plan for scale-up. So for those of you who don't work in the rare disease space, and in fact, before G2MC, I probably could say that would include myself, in thinking about you know, what kinds of genomic medicine were really ready for prime time in terms of evidence-based and clinical utility, thinking 5 or 6 years ago, exome sequencing for patients with rare undiagnosed diseases was really the low-hanging fruit. And I guess this field started back a little more than a dozen years ago now with this pivotal report of a 5-year-old child with extensive gastrointestinal issues, presumed to be inflammatory bowel disease, had over 100 surgeries in the first 3 years of her life. Initial conventional molecular analyses were all negative, and sort of the first, at least high-profile report of whole exome sequencing in this child identified a point mutation in ZIAP. It's known to cause immunodeficiency and colitis, and based upon that molecular diagnosis, an intervention was made, bone marrow transplant, and essentially a cure for this child. At 42 days post-transplant, the child was able to eat and drink, and there's been no recurrence of GI disease. So since that time, really whole exome sequencing, and even beyond that now, genome sequencing has become the standard of care in Western countries for the diagnosis of patients with rare undiagnosed diseases. And through exome sequencing now, there has just been a proliferation of understanding genes for monogenic disorders. And as of about a month ago in OMIM, there are now close to 7,500 diseases and phenotypes for which a molecular basis is now known, most of them monogenic disorders, and now the vast majority through exome sequencing. So this is the background behind why we began the G2MC Rare Undiagnosed Disease Flagship Pilot Project. We were able to get funding from the RTW Foundation to kick this off. We had funding to open 6 sites, G2MC sites across the world in Mexico, Chile, South Africa, Sri Lanka, Malaysia, and Nepal. The University of Maryland and G2MC served as a coordinating center for this effort. And we had funding to sequence 3 cases, actually 3 trios per site, total of 18 trios across these 6 sites around the world. We first sought the IRB to ask questions about what we were doing and the ethics thereof. The University of Maryland IRB deemed what we had designed, as I'll show you on a subsequent slide, as non-human subjects research. And Rich, I checked this, it is non-human subjects research, not exempt. This is because the consent and the results disclosure for clinical sequencing was going to be performed by each of the clinical sites. No one other than the clinical site directly interacted with the patients, and the only sharing was a sharing of de-identified data only. So this is all by design, and as a result, non-human subjects research by US IRB standards. Inclusion criteria for choosing these 18 trios included a proband with a suspected monogenic disorder, both parents available for testing. We wanted to sequence trios to potentially increase our yield of molecular diagnoses. The group had to agree that the case was appropriate for exome sequencing during a pre-review case conference. And some of the criteria for that included the reasonable possibility of suspected disorder would be detected by exomes and exome sequencing, and those included things like the fact that they were multiplex families, there might have been consanguinity, phenotype might have been one that was amenable towards a molecular diagnosis by exome sequencing. And in addition, no other genetic or non-genetic testing would be useful or thought to be useful for the diagnosis. These case pre-review conferences were incredibly rich. It was essentially an international showcase of clinical geneticists that got together via Zoom and would listen to these case conferences. We used a PowerPoint template in which these cases were presented in 10 to 15 minutes with a lot of discussion ensuing. We reviewed the demographics, the medical history, review of systems, physical exam, developmental history, family history, and of course, all of the genetic and non-genetic testing that was performed to date. And then there was team alignment around whether to pursue exome sequencing in that particular proband. And again, these conferences were just so rich. As a non-geneticist myself, I learned an awful lot, but I think that all of the clinical geneticists that were in the so-called room learned a lot together as well. The exome sequencing was performed at Centogene, a clinical laboratory. They reported back pathogenic, likely pathogenic, and variants of unknown significance in a clinical report. The site clinicians also were able to request the raw exome sequencing data from Centogene so that they could engage in their own analyses at each of the clinical sites. And the grant also paid for access and training to Fabric software that was available to each of the sites for their own analyses. And of course, one would want to know whether the internal analyses were able to replicate findings in the clinical report from Centogene, and also provided the opportunity to explore and to look for potentially other variants of significance to the patient's diagnosis and care. And some of the sites had their own homegrown software in which they were able to utilize that sequence data as well for analysis. And then we would have a case conference to review the genetic data in the context with clinical data. Again, very rich conferences in which we were able to make a number of molecular diagnoses and also recommendations for follow-up care in many instances. So this is a summary of the results. You can see that the probands were 2/3 male, 1/3 female. I don't think that deviates statistically from about 50-50 and a sample size of 18. The phenotypes that were present in these individuals varied. We had multiple congenital anomalies, neurodevelopmental and neuromuscular disorders, skeletal disorders, and the like. And you can see that we were able to make molecular diagnosis through exome sequencing in 28% of the probands. Another 44%, we had a suspected diagnosis based on the exome sequencing, but more analysis was needed. Some of that involved essentially the fact that we don't know what's normal in many of these populations, and variants of unknown significance may very well turn out to be pathogenic in those populations, and further investigation of many of these variants are ongoing. And like many places, some of these patients also provided exome negative results, essentially no diagnosis. So again, just to make it real, I'll present a couple of cases. This is a case from Mexico, a 5-year-old Mexican of mixed ancestry patient referred for developmental delay, autism spectrum disorder, speech delay, relative macrocephaly, dysmorphic features, sleep disorder. Exome sequencing in this patient revealed a heterozygous mutation in a gene called ERF, de novo, designated as pathogenic, and this made the diagnosis of craniosynostosis type 4. And now with that diagnosis, much more confidence in follow-up and how to follow this patient. And I'm told that the treatment or follow-up in these patients is to monitor their for elevated intracranial pressure, also speech therapy and educational support. South African 10-year-old female, Sosha ancestry, referred for spastic diplegia, muscle weakness, and dystonia. Exome sequencing revealed a likely pathogenic variant in a gene called GCH1, which is a GTP cyclohydrolase 1 deficient dopamine responsive dystonia. It's known to be autosomal dominant, penetrance female, greater than male. Childhood onset, had checked all the boxes for the likely diagnosis. And here, of course, follow-up, knowing molecular diagnosis was to continue dopamine agonist, since that was a treatment known to be efficacious in these patients. And the father was referred to neurology, discussion of 50% risk to his siblings. Lastly, a 5-year-old male from Nepal referred for muscle contractions of the face, tightening limbs, muscle twitching of the face and limbs, clinical diagnosis of myotonia congenita. Here, the exome revealed a homozygous mutation in HSPG2. Actually, this was a variant of unknown significance that was reported. Sister had similar symptoms, and this provided us enough confidence to actually label this as a likely pathogenic mutation, a cause of Schwartz-Jampel syndrome type 1, and is consistent with the phenotype. So those are some examples of what we found. Lessons learned were really productive and really important in how we begin to plan the scale up of this pilot project. For one thing, as I've mentioned, variants of unknown significance were really frequent in these patients. Maybe not surprising, because these patients were sequenced from populations that are genetically underrepresented in current databases. So we really need population-specific controls to better analyze these exomes. Microarrays, we hadn't really anticipated that in some patients, actually microarrays, in some places, microarrays are not the standard of care for these patients. And in some cases, we had funding to add microarrays to the molecular analyses. As I mentioned, really strong educational and clinical value to these international pre- and post-sequencing case conferences. They need to be continued, they need to be expanded. We need to provide access to those conferences to others. As I mentioned, exome sequencing led to diagnoses in some, but not all cases. And we did be thinking about the next step and the next step. And now more and more, we're seeing reports of whole genome sequencing, RNA-seq, long-read sequencing in exome-negative patients, and we would love to build some of that into the scale-up. Only a few sites use the Fabric software in-house exome interpretation, and we learned that in many of these sites, there's just not the expertise, the personnel, or the time to actually do analyses in these clinical settings, and again, something we need to consider as we plan the scale-up. And further workup, deeper family history, imaging, biochemical testing, and the like would be required for those equivocal whole exome sequencing cases. And so these molecular analyses are just works in progress. And we hope that we'll have more diagnoses as the cases unfold and additional workup occurs. And lastly, but really importantly, we learned that each site is its own site, and they have their own issues, their own workflows, and we needed to provide each site with the autonomy to utilize the limited resources that we were able to provide to them for their own uses, to capacity build, and ultimately to execute the project, bringing exome sequencing to their patients. So in terms of ongoing and future plans, we have a number of publications that are currently in progress that reports this pilot study. We have a study design pilot project manuscript. Also, we were able to convince Topics and Frontiers in Genetics to have a special issue of rare disease research and diagnosis in low and middle income countries. We're going to be submitting case reports, but also this special issue is open to anyone who has case reports or other manuscripts that they would like to provide around this topic. And we're going to submit pathogenic and lightly pathogenic variants to ClinVar and novel population variants to DB-SNP. And as I mentioned, we're really excited about the scale-up. This is a schematic of kind of how we're envisioning this. We have now, again, all around G2MC and meetings that have occurred over the last dozen years, now 22 sites that are interested in this scale-up. You can see they come from all over the world. The 6 sites that we've already engaged are shown in green. We envision a coordinating center of sorts and then the multiple clinical sites. We're going to be working with other organizations that are working in this space of rare undiagnosed diseases internationally. And we're interested in engaging a sequencing partner who will be able to sequence these genes. And in aggregate, we think that over the course of about 3 years, these 22 sites may provide as many as 30,000 samples, 5,000 patients a year, trios, and we would very much like to sequence at least 1,000 population controls to assist in interpretation. So with that, I'll conclude. I'm really proud to be here representing a fantastic team of collaborators and funding from the RTW Foundation as well. Thanks.
Thank you, Alan. We'll take the questions at the end. Let me now invite Vinod Scaria. Vinod is coming here for the first time to GSUMC, but he was part of the team which sequenced the first Indian genome. And then thereafter, we collaborated with him and sequenced the first Sri Lankan genome. That was way back in 2009-10 period, so we go a long way. Vinod is going to talk to us on genomics of undiagnosed diseases, personal to population, and back. Over to you.
Thank you, Vachira, for the nice introduction. It's a pleasure to be here and it's the first time at the G2MC. Over the next 15-odd minutes, I will talk about the genomics in India and how we have taken from one single genome, the first personal genome, to precision medicine in terms of looking at undiagnosed rare genetic disorders. While the slides come up. Sorry for that. you very much. Yeah, so I recently moved out of the CSI Institute of Genomics and Integrative Biology, and all the work that I'm going to talk today was done in my previous institution with a fantastic set of colleagues, many of whom have actually moved ahead in their careers, and a fantastic set of clinical collaborators from not just in India, but across the world, and consistent funding from CSIR, which made this all possible. Now, we started almost a decade and a half ago, and that was the time point in 2008 when we started looking at genomics, and that was essentially the admixture plot of the world, and you had 6 genomes at that point of time, and what you see here is a large gap in the middle, which essentially states that 1/3 of the global population today did not have a genomic representation. That's what we wanted to really cover. And we completed the first genome and announced it in 2009, which was announced in the Parliament of India, with a fantastic set of clinical collaborators and researchers at IGIB, including Dr. Sridhar, who's been a colleague and collaborator. We did not really stop there with the first Indian genome. We went ahead and sort of looked at the entire panorama of genetic variation across Southeast Asia and the Middle East. We had collaborated with Dr. Wajira with the first Sri Lankan genome, the first Malaysian genome with Professor Mohammad Saleh at the University Technology Ammira, and of course, collaborated quite extensively across the Middle Eastern and the Southeast Asian populations to also create one of the early data repositories for populations across this part of the world. And very recently, we engaged with the Indian government with a large-scale pilot genome program called the Indigen. I'll talk briefly about that in a few slides from now. Now, of course, India is a very, very large country. To put in numbers, we are 1/6 of the world population, and that would roughly mean that every rare condition, 1 in 1,000 or 1 in 10,000, would have tens of thousands, if not millions of individuals. Now, this large population is not very homogeneous. They're very diverse, and roughly that corresponds to around 4,000 endogamous defined populations. And that would necessarily mean that India is not just one country, it's around 4,000 different countries put together. But significantly different from the rest of the world, they still have a close-knit family structure, and that means that every individual on an average can trace at least 3 to 4 generations of their pedigree. And all of genetics actually starts with a pedigree. Unlike the rest of the world, we have been largely undisplaced by any of the global major events like war or famine. And that would largely mean that some populations could trace their ancestry as long as 54 generations. And I'll have one example for that. So in 2015, we started a formal program. We had worked on rare genetic diseases before, but a formal program where we could work with clinicians in a formal way of consenting, sequencing, analysis, interpretation, and return of results. And over almost 5 years, we have built it to one of the largest programs in this part of the world, encompassing today and working with close to around 300 clinicians from around 70 major centers across the country. And just before I left, our large repository was around 10,000 patients with undiagnosed rare genetic disorders. Now, I'll just give you one example of the many examples on what we do. Now, this was a family who actually petitioned the president of India to euthanize 6 out of their 8 kids because they have 6 children without an undiagnosed genetic disorder, a very progressive neurological disorder. And the clockwise direction from the youngest to the oldest, you would see that progressively they become deteriorated and end up in a vegetative state. Now, for most of the program, the diagnosis ends the story. And of course, in this particular case, we did diagnose this condition as cystic megalencephaly, in other words, megalencephalic leukoencephalopathy with subcortical cyst type 1, a long name, an extremely rare genetic condition, but exactly 6 children with this extremely rare genetic condition. Now, for most of the diagnosis or clinical workup, this is the end of the diagnostic odyssey, and people forget about these patients. We did not. We actually went back to the community and the population. This belonged to the Nalband community residing in Agra. The forefathers came with the Mughal invaders into India a few generations ago, and they have a written history and tradition of keeping a pedigree, and they could trace to 54 generations of their pedigree. So we went back to this extended community. We did a carrier screening for this otherwise extremely rare genetic condition, and what we found was even more remarkable, that 28% of the population were carriers for this particular rare genetic mutation. So this is an example of a founder mutation in an endogamous or a consanguineous community, which could roughly mean, and these numbers would roughly mean, that 1 in every 400 children born into this community would have this disease. So we went back to this community, educated this community, and we found another 4 children with the same genetic mutation, the same disease. So that would roughly mean that you could use genomics and genetics to now go back to the community, educate the community, and prevent the disease from happening. And for that, you need to have an accessible, affordable genetic test, not a genome sequencing. And that's what we did in 2018 with the publication. And today we have a diagnostic in the market which characterizes the two founder mutations in India for this disease, one in the Nalband community and the other one in the Bansal community. Now, of course, there are many such stories, hundreds of such stories which I can tell. But of course, for people who are interested, there's this paper that you can go back, published in 2019, where we have suggested how we can take from a research program, which is Guardian, to education, to diagnosis, to implementation. And fortunately today, this provided the very evidence for implementing a rare disease policy in India. Now, when you talk about genetic diseases, I don't call them rare diseases anymore. The real point is, how do we know what is prevalent and how do we know what do we need to work upon? And this is very important in a public policy perspective because governments are interested into looking at genetic diseases which are treatable or manageable, right? And how do we know what is treatable and what is really prevalent? One way is to essentially go back and ask clinicians on what do you see in the clinic. And you put in 5 clinicians into the room and you can absolutely never arrive at a consensus. And that's a matter of fact. And the only approach that you can actually come back to evidence-based policy building is to essentially go back to the community and ask a very simple question. Given a community structure, what are the prevalent genetic variants which are pathogenic in nature? And given the frequency of these genetic variants, can we go back to educate clinicians on how can we diagnose them better? With the idea that can we diagnose treatable, preventable genetic diseases. So that's what we did. I have just one example on how we do that. In 2019, we published the first pilot-scale whole genome sequencing of cosmopolitan Indians, around 1,000 odd individuals. And of course, for 1.4 billion people, 1,000 individuals is essentially a very, very small number, right? But nevertheless, this can provide very useful insights into what is the prevalence of genetic diseases in communities in India. I'll give you just one example, and this is from the Government Medical College in Calicut. From the population scale genome data, we did understand that there is a specific condition, primary immunodeficiency disorders. It's a large class of genetic disorders encompassing around 400 genes, very prevalent in populations. So we worked with the local government to establish clinical support, educated clinicians. We started diagnosing patients, and in 4 years, we have created a large cohort of around 200 odd individuals for this genetic condition. And much more importantly, if you diagnose these children very early at birth, you can actually treat these conditions because they're true curative treatments. One is, of course, to offer them a hematopoietic stem cell transplant, which the government supports for free. And of course, patients could avail immunoglobulins on a monthly injection, and this is also supported by the state. So in other words, you can diagnose patients very early on, not with an exome sequencing, but with a very specific genetic test, because in communities in Southeast Asia, you can identify founder mutations, and founder mutation screening could allow us to offer them accessible, affordable genetic diagnosis. And today we are at the verge of actually starting a newborn screening program, because if you can screen every newborn in the population, you can save every single child who would be born of primary immunodeficiency disorders. The next biggest thing that you could really think about is how can you really screen for treatable intellectual disabilities? And today, by the evidence goes, there are around 81 genetic disorders encompassing around 104 genes which are treatable or manageable. And if you can diagnose them very early on, you can put them on very specific therapies. So you can go back to genome scale data and ask a very simple question. What is the prevalence of pathogenic mutations for these genetic disorders that you can identify and therefore influence public policy in building up newborn screening programs? And of course, you have the numbers in there. One of the most commonest is approximately 1 in 40,000 individuals, that is ATP7B, that's Wilson's disease, a prevalent underdiagnosed genetic condition. And we are today working with governments to see whether we can implement, at least for the prevalent genetic disorders, newborn screening programs. Now, when we talk about collaboration across spectrum, not just about clinicians, not about researchers, but also taking it across the world, what we need to keep in mind is that genomic data doesn't move at the speed of the internet. It moves at the speed of trust. So you need to create an engagement policy, not just in terms of understanding genetic information, not just interpreting genetic information, but also community-based guidelines of trust and systems based on trust that people across the globe can work together. And I think the most important and the largest gap in the field is education. And I would not really underscore the fact that you can really change a lot by just educating people, educating clinicians. And over the last couple of years, you have actually created an enormous set of resources. And thankfully, I could see a number of participants in here who have taken some of these courses on very specific areas that could educate clinicians on very specific competencies. I think I'll stop there, but before that, I can say that you could take from a research program looking at population scale genomes, looking at very specific genetic conditions to the Guardian program, engaging clinicians in education and awareness, and offering them very high-end tests, which is in a different program as a clinical service called GOMED. And doing this over and over again, you can actually create value for the industry. And many of these diagnostics have actually moved into the industry. And of course, this could accelerate research in genetic diseases and implementation. Thank you very much.
Thank you, Vinod, for a fantastic talk. Over to my co-chair. Okay.
Thank you, Vinod. Our next speaker is none other than Michele Ramsay, and she comes from South Africa. For those who attended this year's International Conference of Human Genetics in Cape Town, she has been instrumental as a scientific chair. So this morning, she's going to give us a talk on implementation of genomic medicine in South Africa. Michele.
So good morning, everybody, and thank you so much for the invitation to speak here at the G2MC meeting. I must say I have learned so much over the past day, yesterday, and all the innovative ways in which people are really thinking about implementing genomics and education in genomics in low middle income settings. So it's a privilege to talk on behalf of a continent, and a very tough thing to do. And I'm really happy that there are so many of my colleagues here in the audience, and you've already heard some of the students speaking. So Africa is a very complex continent. It covers about 20% of the land mass, and it has 1.4 billion people, 18% of the world population, and over 300 different ethno-linguistic groups. So a lot of diversity in many different ways. But when we think about health in Africa, Africa is still a continent disproportionately burdened by infectious diseases. So it's no surprise that research has really focused on HIV/AIDS, TB, and malaria. But what we are seeing also in Africa is an increase in the prevalence of non-communicable diseases. And this is because of increased urbanization and that epidemiological transition to a more Western lifestyle. So in general, in African countries, there is poor healthcare infrastructure and a low skills base. And this is one of the challenges that we have, that there is a real scarcity of genomic research data on African populations. And we know that we need, because of this expected population growth, solutions that are tailored to Africa. So Africa is the continent where anatomically modern man emerged. And that was, 200 to 300,000 years ago. And you can see that there's been a lot of migration across the continent, of course, leading to increased diversity. And this is our challenge in Africa, the enormous genetic diversity that we see, but also the other layers of diversity. Now, there's an initiative which is about 2 years old now, which is the EU-Africa Personalized Medicine Consortium. And they've done an assessment to look at the readiness of African populations for genomic research, never mind implementation of genomic services. And what you can see is that the countries that are furthest along the way are South Africa and Egypt. And then you can see across the continent, there's very variable readiness for doing genomic research. But I thought to start with just a little timeline for you. So the first prenatal diagnosis by DNA analysis was done in 1984, long before PCR was invented. And in 2003, we started the African Society of Human Genetics, which now has grown such that we have 8 or 9 country-specific societies of human genetics, and we get together every year or 18 months, have lots of education programs, and you heard from Vicky Nambawari earlier about some of the work done by the society. Interestingly enough, in 2011, We had the first African government-supported genome program, the Southern African Human Genome Program, which published its first pilot study in 2017 based on just 24 whole genome sequences. And then, of course, we have this flagship project for genomics in Africa called the Human Heredity and Health in Africa, or H3 Africa Consortium, which has really built capacity and networks across Africa for genomics. In 2020, we published on the first 320 odd whole genomes and just highlighted the extraordinary genetic diversity in Africa. And now, of course, we have this WHO report that you heard from yesterday from Jeremy Farrar, and we're hoping that we can also encourage African governments to support genomic initiatives. Very happily, with the publication earlier this year of the draft Human Pangenome Reference, is that over 50% of the participants had some African ancestry, and some of them even from continental Africa. So we are beginning to build those pan-human genomes for different populations, and that was touched on yesterday. So we have two new initiatives. One is called African Genomic Centers of Excellence, which is still aspirational, but we hope will make a difference. And we also have an African Population Cohorts Consortium to build large cohorts across African countries. So I thought just to divide this talk into Mendelian and then common complex disorders. We've heard a lot about the former, not so much about the latter and what needs to be done, but just two very brief examples. I don't think one can talk about genetics in Africa without talking about sickle cell anemia. And as you can see, about 75% of the cases of sickle cell anemia occur in Africa, with a carrier frequency across equatorial Africa of between 10 and 40%. So every day in Africa, 1,000 children are born with sickle cell anemia. And that means that on the continent, there are 80 million affected individuals. So in a Western setting, you would have access to prevention, early diagnosis, effective treatment, and even cure. And as you see here, Victoria Gray was one of the early recipients of genome editing. And she's African-American, she has sickle cell anemia, she has now been cured through ex vivo genome editing. But this is not something that will be available in Africa. The cost of her editing was well over $1 million. And the question is no longer, how do we improve access to this very expensive gene editing? It's how can we adapt the technology to make it more affordable and accessible? Ex vivo gene therapy or gene editing requires transplant facilities. Those are only available in Africa and South Africa and Egypt. And therefore, you couldn't use that approach. So it's very encouraging to see people now using in vivo approaches for gene therapy. Still a long way off, but it's coming, and that will be more tractable in low middle income countries. The next example is childhood kidney disease. So very early on, it was observed that children with kidney disease from different ethnic backgrounds have different responses to steroid medication. And particularly Black children were resistant to steroid treatment for their kidney disease and would very rapidly progress to kidney failure. So there is now a mutation in the Potosin gene, which we know is associated with a diagnosis and a prognosis for childhood kidney disease. So what you can see in that Kaplan-Meier plot is that steroid-sensitive children respond well and have a good life expectancy, whereas those who are resistant do not do well. And specifically, if they have this mutation in the Potosin gene, their outcome is very poor. So there's utility in this kind of test in that it can lead to treatment guidance, it can be a prognostic indicator, and it's also useful to families in terms of reproductive options. And very happy to say that this has now been implemented as a diagnostic test in the National Health Laboratory Service. So the message that comes across from many of the talks is that you need to understand your population and you need to understand the genetic basis of disease for it to be helpful and useful. We cannot use what has been developed in European populations in African populations before we check whether it actually makes sense. So we see an enormous amount of allelic heterogeneity for conditions like cystic fibrosis, Gaucher disease, familial cancers. But we also see heterogeneity at the locus level. So for instance, for Huntington's disease, there are 2 different loci in African populations that cause this phenotype. We see it for albinism, Fanconi anemia, deafness, and so many other conditions. So we really need to do the research in African populations to be able to implement precision medicine approaches. So I'm going to transition now to complex multifactorial disorders, because these are the ones that are also so very common. And there are a number of things about African genomes that are relevant here. First of all, we see a lot more genetic variance in African individuals, about 1 million more than we do in Asian or European populations compared to the reference genome. What we see is that there's very little data in that the genome-wide association study participants are very seldom African, and if they are of African ancestry, they're usually African American. And then when we look at transferability of things like polygenic risk scores, if the risk scores are developed in European populations, they translate more poorly the further you are genetically away from European populations, and therefore the least well in African populations. So because of this, we've had to develop our own genotyping arrays for African populations and GWAS. We've had to look at effective imputation panels, and we've had to develop pipelines suitable for doing genetic association studies for complex diseases. So we see a lot of variability even within country. And in South Africa, we have many different language groups. You can see how they were traditionally spread throughout the continent. And when we look at them genetically, we can see that there are some distinct differences, most of which are not going to be medically relevant, but some of which may be. So for example, if we look at PCSK mutations that are associated with LDL cholesterol, we can see that across different ethnic groups, the frequencies are very different, and the same with the APOL1 allele, which is associated with kidney disease. So this leads me into a population study across Africa on kidney disease, and specifically looking at regions in West Africa, East Africa, and South Africa. And what you can see in this particular study, which is in older adults, is that the prevalence of kidney disease is over 10%, but it does vary according to region. And as you can see here, there are important sex differences as well. So when we look at the association of APOL1 variants with kidney disease in Africa, the picture is not clear at all. So we saw no association with EGFR, which is one of the measures of kidney function. But when we looked at albuminuria, we saw that there was an association between APOL1 variants and kidney disease. I'm just asking IT. I can't seem to be able to advance the slides. Oh, there we go. So what many of you who are familiar with the literature on the APOL1 risk variants is there is a G1 and a G2 risk variant, and they don't have equivalent function. And what is very important through the research that we've done is it's particularly the G1 genotype that predisposes individuals to albuminuria. So in July this year, there was a study published on coronary artery disease and the use of different polygenic risk scores to best predict disease. And what was done here was to develop a risk score based on information from diverse populations. But of course, there's not a lot of data from Africa. And what you can see here is that the multi-ethnic score, which are the ones that are shown in red, and then applied in target populations of African ancestry, European ancestry, and Hispanic ancestry, and South Asian ancestry, is that although the multi-ethnic polygenic risk score was the best predictor, it predicted most poorly in African populations. Just kind of thinking, where do I actually point this to get it to advance? And the next one. Thank you. So you can just see that it predicts most poorly in African ancestry populations. So the next slide shows the contribution of variants that were identified in different population ancestries. And what you can see here is that African populations are making a very small contribution to what we're seeing. And the reason is that there isn't enough data on African populations. So when we think about the future of polygenic risk scores for complex diseases, we all understand now that the predictive capability is below what we expect in terms of the heritability of a trait. We know that accuracy varies according to trait. And what we know is that if we want to implement polygenic risk scores for population stratification, this is going to be very costly. And we know that there is lower power of these risk scores in non-European populations, specifically in African populations. But this, of course, is also an opportunity because of the tremendous genetic variation. And then what we have learned is that integrated scores using genetic and non-genetic factors are more predictive. And after the session, one of our young students, Michele Camp, will be giving a presentation on her work in our African populations on these integrated risk scores. And you'll also hear another presentation from Caitlin Wheeler, who's also from the University of the Witwatersrand in Johannesburg, talking about a pharmacogenomics application for liver transplant patients. So what are our key messages in terms of genomic medicine in Africa? So first of all, we have enormous opportunity because of the high genetic diversity and the low linkage disequilibrium. But we also have this very complex population structure, which makes it appropriate that we should have different approaches in different parts of Africa. We have this enormous potential for novel discovery. And we know that whatever we do in Africa is relevant to the rest of the world, not only the African diaspora, but everybody, because of the origins of our species in the continent. We don't have nearly enough data, and we need to increase the diversity of genetic studies to also include many African populations. So I think we all realize in this room that it's not all about research, it is about implementation. And we've heard some really wonderful stories of implementation. But I think in Africa, we particularly struggle with that top one, which is having the buy-in of our governments and the political will to support precision medicine. But we also don't have very strong health infrastructures, both from a diagnostic laboratory point of view and clinical services to support precision medicine. So there are many ways that we can manage this, but we have a lot of work ahead. So I think this statement applies to all low middle income countries. So we're saying to improve individual and public health outcomes through genomic medicine, genomics research must be done in African populations, led by African investigators. But we can't do this alone. And this is why I'm so happy to be part of this community. Thank you so much.
Thank you, Michele. Wonderful talk. Next up, we have Matt Brown, who's the Chief Scientific Officer of Genomics England, and he will be speaking on the implementation of genomic medicine in the Genomics England. Thank you.
All right. Thank you for the opportunity to present. So I'm going to talk today about a bit about the history of the Genomics England program and about how it was involved in the design and development of the UK National Health Service Genomic Medical Service, how it brings together research and healthcare to try and improve healthcare provision, and lastly, to talk about some of the research programs that we've got to try and address issues with healthcare, with genomic medical services, which I think are of relevance to services around the world, albeit this is a first world program. So just the 100,000 Genomes Project itself, most of you will be familiar with it, but for those of you who are not, it was established in 2013 by the then Prime Minister of the United Kingdom, David Cameron. At the time, remember that the largest global genomics program was the 1,000 Genomes Project. So this was a huge step up in terms of what it was, in terms of the numbers the genome sequences that were planned to be performed. But perhaps more importantly, it was a program where it was focusing not on population genetics, but on disease genetics, particularly rare diseases and cancer, with return of results to clinical services and to patients. And that was an additional level of complexity, which does make it quite a unique program. At the end of the day, we sequenced about 110,000 whole genomes that were made up of patients predominantly with rare disease. You won't be able to see the different rare disease categories there, but they were not They were recruited basically by first comers rather than in a systematic approach to try and get all different types of rare diseases. So they're not fully representative of what's seen in a clinical genomics service, but they do cover a wide diversity of what clinical genomics services do see. And secondly, we also did a cancer program which involved tumor normal sequencing of, again, a whole diversity of cancer types, with all of the sequencing done by whole genome sequencing. So that program finished in 2019, and from it, basically, the National Health Service's Genomic Medical Service was established. And that's a partnership between Genomics England, Illumina as the sequencing provider, and NHS. So the NHS provides very much the clinical and patient side of the service, where they identify patients who might benefit from sequencing, do the consenting and obtain the sample collection, and then additionally do the DNA extraction. The extraction is then passed on to a centralised sequencing facility at Illumina's facility in Hingston in Cambridge, where whole genome sequencing is performed, and then all of the subsequent analysis is performed by Genomics England with annotated, interpreted results and sent back to the National Health Service so that they can return those to patients. So this is a combination hub and spoke setup where we have hub for sequencing and analysis and spoke being genomic laboratory hubs, of which there are 7 around the United Kingdom, which actually provide the local interactions with clinicians, both in terms of test ordering, sample handling, but also in terms of return of results. And I think that model is really important as one of the major reasons why this program's been successful, in that you have the efficiency of high throughput sequencing factories and analysis, whereas you also have the benefit of the local interactions with clinicians, both in advising about test ordering and about test interpretation. So there's funding, central funding for all of this whole genome sequencing through the NHS, and there's public listing of all of the different tests for which you can actually order testing. This is available to anybody in the audience here. And so there are currently over 50 rare diseases for which you can have whole genome sequencing testing ordered, and a bunch of cancers that you can see listed on the slide. This national test directory is updated every 12 months based on global evidence for the utility of whole genome sequencing to provide some clinical benefit. Now, as I mentioned, there's a very strong link between research and the clinical side of our services. So on the healthcare side, I've explained what happens there, but then what I didn't mention was that all patients who are recruited in for clinical sequencing are asked to provide consent for their data to then be made available for research purposes. And that data is then linked up to an extensive data linkage network that we have in the United Kingdom. Over 90% of patients who are asked to consent do provide consent. That data then gets deposited into what we call the National Genomics Research Library, which is a huge repository of clinical and genome sequence data, which is then made available in what we call a trusted research environment, where researchers from around the world can look into and analyse that data, and through specific controls about what data gets released, can return that data, can use that data for research publications. And so that data is freely available to non-commercial researchers around the world and has been since 2019. It's a phenomenally valuable resource. This is just looking at what the National Genomics Research Library actually contains. Currently, it has around about 200,000 short-read whole genomes in it that cover predominantly rare diseases, but also cancer patients, and an increasing amount of general population data, which comes from studies that we've done, for example, a 40,000 whole genome sequence study of COVID, which is available in the research library, but a whole lot of other cohorts as well. And the numbers are progressively increasing, so that by 2026, we expect to have over 500,000 patients, let alone whole genomes in the data set. It will also become increasingly relevant to, although it's predominantly a disease-focused cohort, because of the amount of general population sequencing that we're doing, it will become more relevant as a general population cohort as well. So going back to the 100,000 Genomes project, as a few people have mentioned today about diagnostic yield from rare diseases, this is actually, I think, relatively low and less than we would have predicted 15 years ago. In fact, in our pilot, we see around about 25% of participants who are referred actually get a diagnosis, but that varies substantially by the disease category, so that some disease categories like as hearing or ear diseases or eye disorders have very high diagnostic yields, presumably because they're relatively monogenic systems. Whereas other disorders, such as, for example, dermatological disorders, rheumatological disorders, and gastroenterological disorders, which by their nature tend to be more polygenic, have had lower success rates. And that has helped shape what sort of testing actually is available by whole genome sequencing on the National Test Directory. We also saw a number of other features which influence the likely success rate. So, for example, the patients that were referred as singletons, such as, for example, patients with later life diseases, had lower success rates compared with those where we had familial duos or trios. And that yield, though, is improving over time as we get better with, for example, being able to identify de novo variants because we have better, larger reference data sets. And when we get better at phasing, particularly through use of long-read sequencing, which we use extensively. So because of our strong links with the research community, we actually get a lot of benefit from the global research community looking at our data and then making diagnoses that have not been made by the NHS services. So this is looking at the number of patients with monogenic diseases for which a pathogenic variant and diagnosis was made by academic researchers during 2022 alone. So in that year alone, we returned over 1,500 putative diagnoses to the genomic laboratory hubs of people who hadn't been diagnosed initially through the short-read whole genome sequencing analysis. In addition to this, we have the ability to do recall by genotype. So we return, we contact, if researchers find individuals in our data set where they want to actually get in touch with the caring clinician and through them, the patients involved, we enable that, and in 2022, we made 600 such contacts with clinicians. So because there's clearly a long way to go in terms of improving both in rare diseases, the diagnostic yield, and in cancer, the quality of the personalised medicine information that you can return to the clinical services. We have a number of new initiative programs, which I'll talk through now, including long-read sequencing, multimodal data sets, including digital histopathology and whole genome sequencing, a major diversity program, a newborn sequencing program, and a multi-omics program. So just briefly, the long-reads program is looking at the utility of long-read sequencing using Oxford nanopore sequencing to improve cancer diagnostics, both by providing rapid cancer, local cancer sequencing in peripheral centres around the UK, but also in providing the epigenetic sequencing data that you get out of Oxford nanopore sequencing for example, neurological malignancies. That will have sequenced roughly 2,000 cancer whole genomes by the end of next year. We have a really large multimodal program which brings together our short-read whole genome sequencing data, digital histopathology from the reference cancer, and any available clinical imaging that was done on those patients. This data set is now 4 times larger than the Cancer Genome Atlas, which up until now has been the largest such multimodal data set. It's housed in a specific compute facility that's suitable for AI-based interpretation, and again, is freely available to academic researchers. Looking at diversity, actually the 100,000 Genomes project is as diverse as the UK general population, unlike other UK data sets like UK Biobank, which are predominantly white ancestry. In our data set, we have the same demographic distribution as the UK population. So what we found through the 100,000 Genomes project was that the diagnostic success rate did vary significantly by ancestry. And some of that was related to the type of genetic diseases you see in different ancestral groups, such as, for example, actually we see quite a high diagnostic success rate in South Asians because those families tended to have recessive or codominant diseases, which are easier to map. Whereas in, for example, people of African or Black ancestry in the UK, the success rate was much lower. And that was a result of multiple things, including the fact that most of the families were either duos or singletons, and also because of the number of variants that ended up being triaged that had to be assessed. So in this figure here, I show you the number of variants that we saw according to ancestry. Of course, the more variants you actually have to assess, that means the harder it is to actually make a diagnosis. And you can see that although in Ashkenazi Jews, because there are relatively few ancestors and variants because it's a less diverse population, in African populations that proportion was much higher, in particular in East Africans, which I think are a substantially understudied group globally, where the difficulty was much, much higher. And this represents an important diaspora in the UK, so it's an area that we're working on extensively. So 2 years ago, we were funded to do an extensive diverse data program, which has quite a range of different initiatives to try and improve and achieve equality in performance of genetic medical services, irrespective of ancestry. It focuses particularly on Black Britons and South Asians. It will have succeeded by end of 2025 in sequencing roughly 25,000 non-European ancestry patients, including cohorts with, for example, 10 to 15,000 sickle cell disease patients. And again, that data is going to be made publicly available in our National Genomics Research Library. Coming close to the end, because I know I'm running short of time, but two other really exciting projects that are kicking off at the moment are a newborns genome program, where we are sequencing over the next 2 years 100,000 newborns in the NHS as a study to look at the utility of genome sequencing in place of current typically metabolic sequencing using the Guthrie card test in the UK. Yesterday, we announced the list of the 223 genes and 500 different diseases that will be screened through this program, which increases if successful, will increase the number that will be screened from the current 9 that are sequenced in NHS England to over 500, and hopefully more as we learn more about genomic antecedents of disease, and in particular because it will only be returning to families findings where the genetic variant points to a disease for which there's a treatment. As treatments for rare diseases improve, more diseases will actually be reported back to parents. And then lastly, and a particularly big effort to try and improve rare disease diagnoses, we have a major multimodal program. We already have sequenced and available in the NGRL roughly 5,000 long-read whole genomes from both European and non-European ancestry populations. But we've recently announced a partnership with Oxford Nanopore, where over the next 12 months, we will be sequencing 7,500 rare disease participants who are either patients who have not had a diagnosis from the 100,000 Genomes Project, or in 2,500 of them where they have had a diagnosis, so that we can work out the relative utility of long-read sequencing in that data set. In addition to that, we've completed sequencing of 7,500 RNA sequencing samples from that data set. Again, that data is shortly to go into the National Genomics Research Library. We've got a partnership with Metabolon to do comprehensive metabolomic profiling of this same 7,500 individuals, and we have a comprehensive proteomic data set of those. So by the end of next year, in the National Genomics Research Library for those 7,500 individuals, you'll have short-read DNA, short-read RNA sequencing, long-read DNA, epigenetic profiling, proteomic profiling, and metabolomic profiling, free of charge to the global academic community. So how do you access that? So Genomics England pretty much established the secure data environment route for being able to access it. You have to submit a proposal to an access review committee, which reviews that it's appropriate. You then have to undergo a research training to make sure that you use our data sets appropriately. We have a trusted research environment which you can work within and for which there are currently no costs to access it or for compute for non-commercial researchers. And then to remove data from it, we have an airlock process, which means that you can only remove de-identified data that meets our patients' consent requirements. So I'll finish up there. There is a contact details for anybody who wants to reach that. But if you have any questions about it, please feel free to contact me or speak with me during the meeting or afterwards. Thank you.
Thank you, Matt. And now we had our fifth speaker from Qatar, and she is going to talk about the implementation of genomic medicine or rare disorders in pediatrics in Qatar. All right, and she is Dr. Ammira Al-Shabeeb Akil. Welcome.
Okay, so thank you so much for the invitation to give me a chance to speak about a very, very underrepresented geographical area, which is, I heard it only once in this conference until now, which is the Middle East. So thank you so much for that. And I will be very happy to take you through a journey of how we established what we are doing now in Qatar, which is a rising star in the area of genomic medicine and also in the implementation of that, especially in the rare diseases and other complex and polygenic diseases. So my name is Ammira, and I'm a head of the Laboratory of Genetic Medicine, and also leading a program for that. And I will take you through a journey that how we started and where we are up to now, and how these building blocks that I'm going to present will give us the understanding about how things is moving in the right way in this area of the world. So this is my talk title. It is about the rare diseases pediatrics in Qatar, the implementation. I'm not going to just speak about this, but I'm going to take you through how we did that and other things as well in my way, just to give you an overview. So I work at Sidra Medicine. I don't have anything to disclose for this talk. The structure of my presentation, it is going to take you through the setup that we have, starting from the leading hospital and healthcare provider in the Middle East, which is Sidra Medicine. I'll take you to the Qatar genome, Qatar population structure, the genome of Qatar, and also the capacity of building the genomic medicine in the country. The objective, it is to understand Qatar commitments to provide support and care for all individuals affected by rare diseases, and that's just relevant to this talk. But we are also care about other diseases, not only the rare ones. So here we go. So I am someone who have been studying in Australia. I graduated from there, and I also had degrees from France. I back to Qatar, the land of the investments in everything good, not only in building great stadiums and carry the best World Cup in the world, but also in building science and scientific committees that can lead everything that you can hear about for the region. So here is the Sidra Medicine, where the place used to build the structure for the Qatari genomes. So Sidra Medicine is a state-of-the-art hospital. This is where I work, and where are all my colleagues who are, I'm talking on behalf, really doing the best for our populations, and that's for the genomics and other diagnostics, and also for very, very important surgeries for children. So it is a reference hospital in the regions, and it is built on the vision of Her Highness Sheikh Al-Mawza bint Nasser, the lead of all the initiatives in Qatar. So the triplets mandates of Sidra Medicine, it's about patient care, medical education, and also about the precision health research. So what we're doing for research, it is implemented in the clinical settings, and that's how we are operating. So what we are doing for research that will take us to the diagnostics in the clinics, we are researching for every patient. So people who are coming to Sidra, around 15K visit per month, they will be phenotypically characterized, and thus will go for different cohorts for polygenic diseases, rare diseases, and maternal health diseases and care. And of course, every other patient, or actually other person who coming to the hospital as a parents, for example, not sick people, they will go for as a control cohorts to be used for the comparison and all of the other things that you can imagine. Of course, with the very big data, health electronic record data that we are also accessing. So the 4 pillars of the precision medicine program established by Sidra Medicine, and I'm proudly a major part of it, have 4 pillars. First of all, we're designing screening strategies. Then we do biorepositories for those patients who screened. We advance diagnostics, and we design personalized therapies. We do have a very much successful stories for that, and we do have a very well-equipped facilities to deliver gene therapies and also for the GMP, for the drug manufacturing grades. So until now, we do have, in addition to Qatar genome data set, which is outcome next after today's, we have a disease-specific cohorts, which is whole genome sequenced. For the Mendelian disorder, which is much relevant to this talk, we have 1,295 currently enrolled, and that's until last Thursday. And we do have 1,128 whole genome sequence out of these monogenic diseases. This population-specific, disease-specific monogenics Mendelians from different clinics in the hospital. And we also have an autism genetic clinical cohort, and that's over 1,000 currently involved, and over 800 of them already whole genome sequenced. Of course, with all the relevant clinical and genotypic data, phenotypic data available beside the whole genome sequencing for each patient. Of course, I'm sorry for the size. I don't know why it looks like that. So this is something like, I think it's repeated here. In addition to the cohorts that we recruiting actively, we established the Genome to Cure, a clinical research, metabolic and genetic program, and that's to translate and facilitate the translations of the research findings to the clinics. Of course, led by clinicians and researchers, and that's under my leadership, and it is designed specifically to align with Sidra research pillars, which is screening, biorepositories, advanced diagnosis, and personalized therapies. Qatar genome is something like I have to stop on, and that's because it is a major achievement for the country and for the region, actually. We started by the announcements of Her Highness Sheikh Hamouza at the WISH Summit, which is the World Innovation Summit for Health in 2013, where she announced the Qatar Genome Program that will lead the way for the cure and personalized treatments in the region. And this is in a very underrepresented population, as I mentioned. It has comprehensive phenotypic data, multi-omics approaches, and also we look for the implementations in the centralized healthcare system and the state-of-the-art infrastructures, which is the, leading the way in the design of the Qatar genome, also in the implementation. Very quick, this is how it is started, and that's on phases, phase 1, 2, 3. We're currently at phase 3, and that's how we could, by using this data, we believe that it will be a first, Africa, as mentioned by professors here, it is the site of the migrations of people to the other part of the world. And we think Qatari population structure, it is representing that migration history. And that's why having a well-established population-specific genomic data, it is essential for the rest of the world, whereas all the people came from Africa, went to the Middle East, and distributed, as you can see. Here's that we started in 2013, was the vision announced. First genome was in 2015, 2018, 10,000 genomes, 2020, 25, and we have now almost close to 40,000 genome release that we can just use, and we have access for international and outsider to Qatar researchers who are collaborating with us on using these data. As I said, the Middle East and North Africa is very less represented, and that's why we took the opportunity of the expat community living in Qatar, and thus there is around 400 to 5,000 millions Middle Eastern in whole the region, many of them represented in Qatar, and that's why we took another initiatives of sequence, whole genome sequence, is expat Arabs, originally Arabs, not just by passport, okay? And we save it as a data for the region also to use. Of course, this is Qatar Genome is one of the few genomes programs in the world which is mentioned by Nature journals, and that's one of the things that we are proud of. Quickly, the structure and the variant distributions of and allele frequency spectrum of Qatar Genome, and this is an article published just like last year, showing the structure of the pathogenic, likely pathogenics, and the novel variants comparing to the one which is known in the genomes, and that's very, very specific for the population in Qatar. And as I said, the population is very specific and it is structured in a little bit different way. We do have three major based on the principal component analysis, three major subpopulations, and the Bedouin subpopulation, it is one of the major representing Arabs, and that's divided into another two subpopulations. The total is 5 subpopulations represented in Qatar, and that's something like very important to know, so to design therapies and also to target certain diseases, and also to be able to build a reference genome that is very much required for any diagnosis. So rare diseases, I am going to start here with some more things to tell you about these diseases in Qatar. Current population of Qatar is almost around 300,000 native Qataris. Rapidly growing community. There's a lot of founder mutations and isolated, because it's isolated and old community. Qatar nationals is 15% of the whole population. Consanguinity rate, very important, is more than 55%, sometimes reach to 70%. Tribal structure, as you can tell from any Arab community, common disease is totally different from Caucasians, as you can imagine. Consanguinity and its clinical relevance and consequences. Very much a lot of information here. We can discuss maybe later, but it is almost recessive alleles with multiple affected members of the families, de novo diseases with each child, and coexistence of multiple genetic disorders in a single family, and also in a single patient. We have pan-tribes versus tribe-specific founder mutations, and we also have homozygous mutations, autosomal dominant disorders all over the place. Familial clustering, of course, Mendelian forms of complex diseases, and very high consanguinity rate, as I mentioned, 23% as a first cousin, 52% as second cousin, and inbreeding coefficient is almost very high, which is, as you see, is 0.237. A very simple example of how the families are complicated. The pedigrees, I couldn't search for more complicated than, but I do have more than this, but this is one of the complicated Qataris, native generations running with Mendelian diseases for generations. Our commitment toward all individuals living with rare diseases is to immediate and future vision. We need to ensure equitable access to this diagnosis, treatment, and prevention, and also to establish comprehensive lifelong healthcare covering screening, counseling, genetic counseling, treatments, and support, and access to all the resources without compromising the quality. So here we have the services for rare diseases in Qatar. We do have premarital, preconception genetic screening. We do national newborn screening. We have a national center for rare diseases in Qatar, and this is have different ways of doing the diagnosis, reproductive genetic counseling programs as well, and population-based platforms, which is Qatar Biobank and Qatar Genome. I didn't mention about Qatar Biobank. I focus on the Qatar Genome, but there is a well-established biobank just running beside the Qatar Genome for the same people who have been sequenced and reported all the other data. We do have educations and research for rare diseases, promoting academic industry partnerships as well. Genetic services, it's focused in two main providers, which is Sidra Medicine, as I mentioned, the place where I came from, and also the Hamad Medical Corporation, which is another government public hospital, very well established for the rare diseases. That's for adults mainly, but ours will go for children. What's the service we're providing? Of course, I focus on the children. We do have for adults, pediatric clinical genetics, metabolic genetics, dietetics, and also cancer genetics, prenatal genetics, reproductive genetics, and also the newborn screening, which is I also will touch base on next. Example of the monogenic diseases that we are just whole genome sequence and return the results to the clinicians. Some of them novel, some of them are well known, and that's all based on our research facility. And we just like able to return the results to the referring clinicians and report back to the patients, and treatments changed, treatment implemented, and that's how we are just currently operating. Total number of babies for the newborn screening, the standard newborn screening, right now 385,000 over. Total confirmed cases 1,600, and the incidence rate of the monogenic diseases 1 in 300, which is so much high comparing to the other part of the world. Very, very happy to see that Qatar Genome newborn sequencing study of Qatar is listed by the Genome England as one of the studies for the newborn sequencing using whole genome and genotypic data in the Middle East, and it is the only one, as you can see. I'm very proud also I'm part of this study. So why whole genome sequencing? I think this is not a relevant slide because everybody knows why whole genome sequencing, for all the reasons that you all know, but unfortunately not everybody can afford doing whole genome sequencing, but we are just lucky to have all the infrastructures and the facility and the government support that we just use to get this all implemented. Newborn sequencing, I want just to stop a little bit here because this is something like just established. We are doing it for research. Currently, in collaboration with the clinical side, this is a pilot program that we are looking for monogenic, polygenic diseases in the newborn samples. We're collecting low blood volumes, we collect cord blood, and that will go for polygenic risk scores, genomic risk scores for certain diseases, and also for the monogenic diseases packages, package 1, 2, and there is other, but the slide is very busy. I'm sorry, it cannot be, I don't know what's happening with the presentation. So there is some work is happening to establish the genomic sequencing for screening in Qatar for the newborn, and that's a first step toward moving toward the whole genome sequencing for all the implementation of the screening using the genomic sequencing for the newborns, and also return the results, and that's something that we're also working on. A very typical genome analysis pipeline in the context of the newborn screening that we are following, starting from the DNA sequencing, of course, and then until filtering the pathogenic variants, and then screening the other newborns. So education, this is something like we do, as you can tell, to implement all what we are doing. We provide academic and clinical education and training for medical and biomedical students, clinical and biomedical genetics fellowships we're offering. We do genetic counseling programs, and that's something like locally established. We also send students to abroad to study genetic counseling because we are just as everywhere else, we need more of them. Medical genetics clinical team, also adjunct faculty in all the clinical, in all the teaching universities. And also we are designing and also leading conferences that is specialized for rare diseases in the region, mainly, and not even more in Qatar. Gene therapy stories for spinal muscular atrophy in Qatar, something like very, very promising. We are very proud to say that we have been treated almost many children less than 2 years old using the gene therapy for the SMA, and that's part of the management at Sidra Medicine. We also extended our services for treatment of children, not only for those in Qatar, but those who are from the region. We have patients from Iran, from Iraq, from Syria, from Spain, from Morocco, from Algeria, from Libya, from Sudan, all treated at Sidra Medicine for this gene therapy. Very expensive, but we are just like take the lead of treating those kids who came to us, to our hospital for treatment. Take-home message, ensuring the early diagnosis, equitable treatments, and prevention to all individuals with rare diseases living in Qatar. And that's something that I'm close by. But before I go, I should show you the people who are working with me on designing the newborn genome sequencing for screening in Qatar, including Professor Robert Green from Harvard and BabySick Project, and my own collaborators, not for the department or whole people, that's all just my own collaborators from outside the country and inside the country, and also our patients who are our advocates as well. Without them, we cannot do anything. They are part of the partnership and we are very proud to help them. And this is my team, okay, who are working, helping me and implementing everything I just spoke about. They are collection of staff scientists, bioinformaticians, research specialists, postdocs, and including our chief research officer, Khalid Fakhro. I think some of you knows him. And before I go, I'm just inviting you to attend our 9th edition of the Precision Medicine and Functional Genomics in November 11 to 14 in Qatar. And that's something like you will be very, very happy to attend because it is seriously the biggest in the region. We are leading since 9 years, and next year will be the 10th anniversary. I would really welcome everybody to join us, and let me know if you need any help in that and if you have any question. Thank you very much.
Thank you, Dr. Ammira. We shall now have the Q&A session, and I'd like to invite all the previous speakers to come on stage and
I'd like to thank the 5 panelists for 5 outstanding, extraordinary presentations. I have one question relating to polygenic risk scores in Africa, more related to Michele. The question is, where do you think we should start? Because the problem is very large, if I understand. The population in Africa is hugely polymorphic. Then it is stratified, as you mentioned, in many subpopulations. Then we might deal with some different environmental factors. And then also, occasionally, the geographical, political borders do not match the ethnic borders. So these are 4 major differences with the European population where it was not so easy to start with. So where should we start with the resources we have in Africa?
Mark, thank you very much. It is, of course, a big problem in terms of not having enough data. I think getting more data is absolutely key. Tomorrow, Shigun's going to talk a little bit about diversifying and including more African populations. But I think until we do more, we're not going to be able to do this. So we need not only the genetic data, but also all the phenotype data that goes with it. And one of our challenges is 54 countries, different, as you say, political systems, sharing data across boundaries is challenging. So I think we have an enormous amount of work ahead of us. I think, we are looking also to industry partnership, because I think that's going to be key in terms of resourcing more genomes. I think our situation is so different to Qatar. But I think there's such potential for discovery that can really help the rest of the world because it is the cradle of humankind. So thank you for that question.
Yeah, so great talks indeed. I agree with Mark. Very insightful, especially touching upon population that we have never heard of. So we are, of course, very well aware of Genomics England, but Africa, especially the differences between the northern part of the continent and the other part. India, Qatar, of course, we're well aware of what amazing things we're doing down there. We heard of PRS, but I haven't heard PRS from Matt. despite the fact that you have a lot of genomes already analyzed. And I am a strong opponent regarding PRS as far as clinical implementation is concerned. So that's where I wanted your comment. So it's something which is really emerging. It's very promising, but is not yet ready for clinical implementation. And honestly, I'm not sure when it's going to be ready. It sounds very attractive. It can sell like hell, but we're talking patients here. So where do you stand?
Okay, so Genomics England, our USP has been about rare diseases and cancer. So I've come from a common disease background, and so after arriving at Genomics England, where I see it being most useful for us is in helping us in the undiagnosed proportion of patients who came into our rare disease program because of a clinical belief that they had a rare disease. But actually, of course, many of those are actually going to have just the severe end of a polygenic disease. And so we're using the PRSs to try and pull that out. That's very much a research program. In the UK setting, the polygenic risk score scene is dominated by the Our Future Health initiative, which you may know of and some in the audience will know of, is on track to recruit 5 million people by the end of, they've got now a bit over 3 more years to go, who will all have a SNP microarray performed and polygenic risk scores calculated. And that is going to give, I think, for developed countries anyway, a really good handle on how useful this is actually in healthcare systems, because I think that's really an unknown. Virtually all of the PRS data that's available so far comes from highly ascertained case-controlled data sets or cohorts and doesn't actually represent necessarily what's going on in the general population where you might actually apply that in a clinical setting. So I'm personally optimistic that it's going to prove useful, but I think I'm going to wait and see for that real-world experiment.
Thank you very much for your presentation. My question goes to all the presenters, and I would like to hear a view of both Michelle and Matt on this. It's about what should be the yardstick that we should use to increase representation. I've had people talk about in my country, so Africa is 1%, so we are going to aim for 1% in Africa, or Qatar is 2%, we aim for Qatar 2%. Should that be the yardstick? And the reason why I'm asking is that we know we've heard a lot about genetic diversity in Africa population, or maybe other populations, South Asian population. It's much more than European population. And the reason why we want to capture all this diversity, because we know that there is more genetic variant in those populations. And I think that what should be our interest is how to capture those variants, not really about the percentage in that population. So what I'm asking is that if Africa has, like, say, 2 million more variant than our population, should we be using a yardstick to capture those genetic variants, or should we be using a yardstick of the percentage of those countries in where they are being captured? I'm not sure whether you got my question.
I'm trying to get your question in there. Can you just clarify the question, please?
Okay, so let me give an example of in the UK, and Matt mentioned this. In the UK, let's say, I'm not sure, I think it's about 1% of African in the UK. So if Matt is trying to enroll people in the UK for genomic study, should he be aiming to enroll 1% of African in the Genomic England, or should Matt be aiming to think about how much of diversity in terms of genetic do we have in African, in Asian, and all that, and use that yardstick to enroll? That will not be 1%, because we know that African, for example, has more genetic diversity than other population. So I'm asking, so should we be using percentage of the population to increase representation, or should we be using the genetic diversity in those populations to increase representation? Michelle, did you get the question now?
Yeah. Okay, so to re-express that, should we be recruiting in proportion to the demographic distribution of the population, or do we need to select our recruitment to the actual research, the use case. And so actually Genomics England, we get a lot of our data through the NHS, which is basically means it's whoever turns up, it's proportional to the demographics of the country. But in our specific research programs, we over-sample effectively in non-European ancestry populations because we feel that we actually need to get to a particular cohort size for us to be able to actually make meaningful contributions. So I definitely think you've got to oversample so that you actually end up with a big enough cohort to actually address what you need to address.
Shuldiner, I think we are lucky that we have this gift of African genomes being sequenced in other countries that we can leverage and use. But I think there's no getting away from doing the research in Africa, considering the environment and the diversity. So I think, what we need to do is the advocacy to get our governments to invest in these kinds of projects. And in the absence of that, I think we're never going to really truly understand complex diseases. We've got to do the work in Africa, but we can't do it alone.
I have a question about newborn screening and whole genome sequencing. For example, Ammira, when you set up the program in Qatar, are you still doing Guthrie cards and biochemical tests, or is everything done by newborn screening? And then I don't know if Alan or Matt or anyone else wants to chime in. Do you think that whole genome sequencing will replace Guthrie cards and biochemical tests?
Very good question. We still do the standards as part of the standard, like part of healthcare. But now we are moving toward the genomic part of the newborn screening. Of course, we are not going to stop doing the standard, unless we are fully implementing the screening using genomics. And also probably we'll run together. Sometimes we need to confirm this with that with this, So I think the answer is, both are will be used. Currently, the standard is running. This is mandatory requirement. And we are establishing the genomic screening for newborns step by step. But all the things is set and we are just into it. Thank you.
Yeah, so my question, this is Dr. Rupesh from Nepal. I need to ask a question to Akil. As you have already mentioned that you have already treated 40 patients with SMA, even in your country and the nearby, is there a chance if I provide some of the patients who has fulfilled the criteria, up to like 2 patients a year, can be treated in Nepal with the Gel-Zosma, what you are providing there?
Can you repeat the question, please?
Yeah, my question is, you have mentioned that SMA patients,
Yes.
in your slides, 40 were treated there in your country, who belongs from Qatar and who are from the nearby countries, like the neighboring countries. So is there a possibility if we have the patient who has fulfilled the criteria of, there are only 20% of the patient who fulfills the criteria for Gel-Zosma. So if we have those patients, are you going to provide us the treatment for those 2 patients?
You know how much it costs this?
Yes, I know,
treatment,
$1.2 million. That's why I ask you. When we are suffering, we are struggling even with the sequencing the patients, and you have already doing that work. That's why I ask if there is a possibility. Anyway, 1 patient a year.
Yeah, I mean, if you notice that all the countries, excluding Spain, they are all from the MENA region, and that's what's part of our commitment. And every patient, there's 40 patients. Of course, there's 16 patient Qataris. The rest are non-Qataris, Every patient have different setup, different story why they ended up in Qatar for the treatments, and they ticked all the boxes to receive this support from us. So there's always a room, and you need just to reach us and see. But yeah, this is have to be tested, of course, by the medical genetics departments, and then the decision can be made. You can talk to me. You have my email. Yeah, thank you.
Hey, are there any more questions? I think, Matt, you were trying to answer something just now, did you? Were you trying to answer a question just now?
Yeah, that was about the newborn screening.
Yeah, so I was actually going to say that a situation that Ammira has reflected in Qatar is exactly the same as what's happening in the UK, that we're continuing going with the current dried blood spot, Guthrie testing, whilst the research pilot goes on with the sequencing study. And if that study over the next 2 years actually performs really well, then I guess people are going to want to look at the dried blood spot screening. But at the moment, they're both going to go forward.
Hi, Matt. I have a question for you again. So, in one of your slides, you showed that in the quad families and the trios, the quad families has the less success ratio compared to the trios family. Could you please comment on that actually?
I'm sorry, you'll have to repeat the question. I really didn't.
So one of your slide, you showed that the trios has like less, is that like more success rate compared to the quad where you have the like more than one sibling, right? So could you please comment on that? Why? Because conversely, you should have the higher rate when you have the, more family members, right? Especially for the disease.
Yeah, so what we saw was that Singleton recruited members of the 100,000 genome study had a lower diagnostic yield than where there was a duo or a parent case trio. And I think that's largely down to the better ability to diagnose de novo variants when you have parental data. As I said, though, that advantage is diminishing as the data sets of healthy control sequences increases and we have better understanding of what's de novo, and the number of the number of de novo variants that need to be assessed per parent case trio is dropping because of that increase in the overall sort of healthy control data sets that we've got. Also, just the more diseases, the more we know about each disease, the more diseases that have been published, like Alan mentioned, the 7,500 that are now in OMM. Actually, about a third of the new diagnoses that we're making are coming about because of new gene discoveries that have been published in the literature. So I personally think that that difference will diminish over time because particularly of those two factors. Also, I guess long-read sequencing has meant that we're better able to phase data independent of whether you've got both parents, and that also is an advantage if you're missing both parents.
Thank you. I think we have come to the end of this session 4. Let's put our hands together and thank all our speakers.